Good day, this is Professor Will, CBMD PhD. And today's Tuesday, April 5th, 2022. It is 5:12 and 35 seconds, AM Eastern Standard Time. And today's research, microbiome. So let's come to it here. New methods for probing the bewildering diversity of the microbiome. Date April 4th, 2022. Source, Arizona State University. The scientists describe a new method for probing the microbiome in unprecedented detail. The technique provides greater simplicity and ease of use compared with existing approaches. Using the new technique, the researchers demonstrate an improved ability to pinpoint biologically relevant characteristics, including a subject's age and sex, based on microbiome samples. Known collectively as a human microbiome, these tiny entities influence an astonishing range of activities, from metabolism to behavior, and play a central role in health and disease. And some 39 trillion non-human microbes flourish on and within us in a ceaseless, independent muscle. And together, they make up over half of the human body cells, though they may possess 500 times as many genes as are found in human cells. Identifying and making sense of the microbial melange has been a central challenge for researchers. The innovative research holds the promise of rapidly advancing investigations into the mysteries of the microbiome. With such knowledge, researchers hope to better understand how these microbes collectively act to safeguard human health and how their dysfunction can lead to a broad range of diseases. In time, drugs and other therapies may even be tailor-made based on a patient's microbiomic profile. And two powerful technologies have been used to help researchers unlock the diversity and complexity of the microbiome by sequencing the microbial DNA present in a sample. And these are known as 16S, a metagenomic sequencing. The technique described in the current study draws on the strengths of both methods to create a new way of processing data from the microbiome. Researchers borrow some of the wisdom that developed from 16S RNA sequencing and apply it to metagenomics. Metagenomics allows researchers to sequence all the DNA information present in a microbiome sample. But the new study shows that the metagenomic approach has room for improvement. The way people currently analyze metagenomic data is limited because whole genome data has to first be translated into taxonomy. The new technique, known as operational genomic units, does away with the laborious and sometimes misleading practice of assigning taxonomic categories like genus and species to the multitude of microbes present in a sample. Instead, the method uses individual genomes as the basic units for statistical analysis and simply attempts to align sequences present in a sample to sequences found in existing genomic databases. By doing this, researchers can get much more fine-grained resolution, which is particularly useful when microbes are present that are closely related in DNA sequence. And this is true because most taxonomic classifications are based on sequence similarity. And if two sequences differ by less than a certain threshold, they fall into the same taxonomic category. However, the operational genomic unit approach can help our researchers tell them apart. Further, the method overcomes errors in taxonomy that persist as relics from the pre-sequencing epoch when different species were defined by their morphology rather than from DNA sequence data. In addition to improvements in resolution and simplicity, operational genomic unit can help our researchers analyze data using what are known as phylogenetic trees. As the name implies, these are branching structures that can describe the degree of relatedness between organisms. based on their sequence similarity. Just as two distantly related species, like worms and antelope, will appear on more distant branches in a phylogenetic tree, so will more distantly related bacteria and other constituents of the microbiome. The most widely used technique for probing the microbiome, known as 16S, ribosomal RNA sequencing, or just 16S, relies on a simple idea. All bacteria have a 16S gene, which is essential to the machinery bacteria need to initiate protein synthesis. The bacterial 16S gene, measuring 1,500 base pairs in length, consists of distant regions, and some of these regions change very little between different bacteria and over evolutionary time frames, while others are highly variable. Researchers realize that the conserved and variable regions of the 16S gene allow it to act as a molecular clock, keeping track of bacteria that are more closely or more distantly related, based on their sequence similarity. Thus, the 8 conserved and 9 variable regions of 16S can be used to fingerprint bacteria. And to do this, a microbiome sample is first collected. This can be a fecal sample to evaluate the gut microbiome, or a sample from the skin or from the mouth. Each body site is home to a different bacterial menagerie. Next, PCR technology is used to amplify portions of the 16S gene by sequencing highly conserved regions. A broad swath of bacteria can be identified, while sequencing of variable regions helps narrow the identity of particular bacteria. Although 16S is an expensive and well-developed method, it has limitations. The technique can only give a general idea of the kinds of bacteria present with limited resolution. In general, 16S is only accurate to the genus level of identification. Enter metagenomic sequencing. This technique sequences the full genomes of all microbes present in a microbiome sample, not just bacteria, as with 16S. Metagenomics allows researchers to sequence thousands of organisms in parallel, providing accurate species-level resolution. The greater resolution, however, does come with costs. Metagenomic data is far richer and more computationally challenging to analyze than 16S data, and more expensive in time and more money to process. The operational genomic technique streamlines metagenomic sequencing while providing even greater resolution. The approach classifies microbes in the sample strictly according to their alignment with a reference database. No taxonomic assignment required. The approach enables researchers to evaluate the degree of species diversity present in a sample. Compared with 16X and standard metagenomic sequencing, the new approach is superior in ferreting out biologically relevant information. By using the classic Human Microbiome Project dataset of 210 metagenomes sampled from 7 body sites of males and female human subjects, the study demonstrates better correction between body site and host sex. And next, 6,430 stool samples collected through a random sampling of the finished population were analyzed using both 16X and metagenomic sequencing. The samples belong to a large, randomly sampled cohort of the finished population, known as FINRIX. The aim was to predict the age of the sampled individuals based on gut microbial composition. Again, the operational genomic unit method outperformed 16X and conventional metagenomic analysis, providing more accurate predictions. New research drawing on still larger datasets will further enhance the resolution of the new technique and expand the descriptive power taxonomy independent analysis. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.